12.3, Chemical Digestion. As we were going through the digestive anatomy back in section 12.1, I did mention some of the various things that occur at different locations as far as digestion and absorption. But we'll spend a few minutes and go over this one more time, and it also gives us an opportunity to review some things that we learned earlier in this semester. For example, recall from Chapter 2, there are 4 classes of biological macromolecules. This would be carbohydrates, lipids, proteins, and nucleic acids. And so certainly we consume and need to consume all of these in our diet. These are all very, very large molecules. Recall polymers, in fact, and they have to be broken down into the individual monomer in order to be absorbed. Another little bit of review, we talked about enzymes earlier. Recall enzymes are very specific. They have a very specific shape, and that shape does determine function. And so these various steps in digestion each require specific enzymes that are in fact located in very specific places in the digestive tract. So carbohydrates are sugars and starches. Sugars are the individual monomers, but these are often found in large polymers. So recall, for example, starch is a long polymer of glucose. The primary nutritional contribution of sugars and starches is energy. And these polysaccharides and even disaccharides must be broken down into that individual monosaccharide sugar before it can be absorbed in the small intestine. This does require several steps. And so what I have here is location, the substrate, the enzyme, and the product. And so first for carbohydrates, if we consume starch, the digestion of that starch can begin in the mouth. There's an enzyme in saliva that is called salivary amylase, and it's going to break down every other bond in that long glucose chain that makes up the starch. And so it breaks it into a disaccharide of only 2 glucose molecules bonded to one another. That disaccharide happens to be called maltose. This can occur in the mouth if we chew our food well and mix that starch with the saliva and give the enzyme an opportunity to work. There is no starch digestion in the stomach, and so any undigested starch is processed in the small intestine. There is a separate amylase, pancreatic amylase, produced by the pancreas, released in the small intestine, and it will also break starch into maltose. But then we have one final step. The maltose has to be broken down into the individual glucose molecules, and this is carried out by an enzyme called maltase, which is also in the small intestine. Protein digestion takes much longer, so it is true that if you have a meal that is high in protein, you feel full and satisfied longer. This is because proteins simply take longer to digest. If you remember in Chapter 2, we discussed the structure of proteins, chains of amino acids, but they do not exist in that extended chain, but rather they are folded into some very complex globular forms. We need amino acids to provide, we need proteins in the diet to provide essential amino acids so we can use those amino acids for our own protein synthesis. Since these are, again, very large, complex polymers, we have to digest that protein, break it down into the individual amino acid that can be transported across the small intestine and absorbed into the blood. Again, a multi-step process. We can begin protein digestion in the stomach, an enzyme called pepsin. And pepsin is just going to start nibbling on this protein. It's going to break down a few of the bonds in this protein, and so the protein will begin to unfold, but it's still some fairly large pieces. So we would call these peptides, still some fairly long chains of amino acids.
he's ready, you know.
And then as those pass into the small intestine, there is another enzyme, trypsin, which will continue the process. It will break down some of the bonds between some of these amino acids, and so it breaks this peptide chain down into some smaller peptide chains, possibly even a dipeptide, which would be two amino acids still bonded to one another. And then we have to have additional enzymes that complete the process. So various peptidases, which will eventually complete breaking this down into individual amino acids. Lipids, or fats, are primarily used for energy. There are a few essential fatty acids that must be obtained through the diet. Whenever you hear the term essential fatty acid or even an essential amino acid, what that means is there are specific fatty acids or amino acids that we cannot synthesize, and so those must be provided in the diet. For dietary lipids to be produced, we have to have the presence of bile, which is an emulsifier that was going to take the fat and emulsify, break it down into some smaller droplets, and that simply allows the enzyme access to those bonds that need to be broken. The enzyme that does that is called lipase. So you may need to review back to chapter 2 to see the structure of a triglyceride, a fat molecule. But the lipase will break the bonds, and the end products would be the glycerol plus those fatty acids.